Deborah, for doing the logistics. I know that's always a hassle. I'm not an immunologist, and in fact, on the form, I'm others, because I'm not a statistician either. So I'm glad you put that there, because I would have had a problem. So I claim to be a systems biologist, whatever that means. And so I try to talk about something that's at least close to immunology to some degree, and it has to do with oxidative stress. So let's see. So I have two parts of my presentation. One is short, even though it doesn't look like it, and it asks, what exactly do we want to get out of these types of models. And then I want to give you two examples. That's the most of this talk. And one has to do with mechanosensing and atherosclerosis, and the other one is a more detailed model of handling oxidative stress and this enzyme NADPH oxidase. So if we look at what exactly we want to get out of it, and you ask systems biologists, or you go through the literature, it crystallizes into two camps. So one camp says we want to understand how things work, we want to make predictions, and we want to find the organizing principles, so the essence of these systems, what makes them tick. The other group says, we want to develop models for personalized medicine. We want to assist physicians and say, this is what you have to do to cure this patient. Now, it doesn't take much imagination to see that number one requires very small models. So you want to prove stuff, and you usually can only do that with small models. And number two requires very large models, because you want to put all the different aspects of a disease in there. And then if you want to personalize it, and there are 50,000 biomarkers, and everything should be accounted for. And so if you like size, you should look at the Blue Brain project that runs on a supercomputer in Geneva and has a column of the cortex, and it's just wow. It's millions of variables. Now, if we look at reality, then most of the models over the last 50 or 100 years are smack in the middle. They are neither small enough to be analyzable really with much, nor are they so big that you can simulate a disease with it. So the question is, are mid-sized models really useful? And I want to go into that and then go into these examples. So if we look generically, what we want to do is we want to take cells or an organ or some other system, like a plant system, and convert it into some diagram. And often biologists do that for us, and sometimes not, and then we have to sort of scrounge for this information. Then we want to convert that into some sort of computable structure. We need to take that structure and put it into some algorithm, and importantly, we want to go back and have insights into these systems. So if we look at the goals, I said the first one is to understand a biological system. Now, if you dig a little deeper, then you say, what does that really mean? What does it mean to understand a system? So one interpretation of understanding is we have all these pieces, and we want to put them together, and if we put them together into a model, does the model still work as we would like it to work, or do things happen in the process? Number two, people often say, well, we want to make accurate predictions of something that hasn't happened yet. And that's what models are for. Otherwise, why do we need them if we already know what the results are? And then three, I mentioned that briefly, is the discovery of design principles, and I'll come back to that. So here's an example of whether pieces fit together. This is a model that we did years ago about purine metabolism. And, poor graduate student worked for 4 or 5 years to get all the information, kinetic information, fluxes, all that stuff. Put it together, didn't work, tested, didn't work, and so on. So you have the cycle that takes a long time. So what can we do with something like that? Well, for instance, we could simulate treatments, and we said, suppose you have too much uric acid, that's at the bottom, and if you are male and old, then you have a good chance of getting gout, and this is the hallmark of it. And on the positive side, you tend to live longer, so you can enjoy it longer. So let's say you have too much uric acid. So the first question is, where does it come from? Well, can we explain it? Well, for instance, we can test with our model now if we increase the activity of this first enzyme, which is a syndrome that you have in this area of medicine, then you can see, yeah, the uric acid goes up. Here's another one. It's a very bad disease. I'm not going to talk about it. Then we can say, can we intervene? And you can say, yeah, for instance, we can block the last step. And yeah, if you block it, you get less uric acid. Big deal. That doesn't take a rocket scientist to figure that out. And then you can say, well, we can actually block it up there, and we can screen the entire model to see whether we have other means of intervention. Then we can say, what are the side effects? Now, these are not side effects like you hear on TV. You know, side effects include drowsiness, headaches, occasional death, and all that. So we cannot do that. But we can look at side effects within the system. So, for instance, again, it's not difficult to imagine. The uric acid goes down, and then the xanthine, which is the last box up, goes up because, you know, it's a side effect. So we can do these things. When we look at design and operating principles, it's a very, very different type of understanding. So here we have two systems, and you could imagine, you know, the top level is maybe gene expression, and the middle level is mRNA production, and then you have a protein that's being generated on the bottom level. And in one case, it's just this cascade. In the other case, you actually have a feedback that goes from that protein or from some product of that protein back to gene expression, like we have, you know, with repressors, transcription factors, and those things. And you can, you know, let's say you observe that this feedback is there, and you say, why is it there? Does it make the system better in some sense? So you can look at criteria of what it means for a system to be better than another system, and you can test these two systems against these criteria. You can do that with operating principles, so you can say, well, if I want to increase the production or the amount of something, I can increase the production or I can decrease the degradation. Is there a difference? Is one better than the other? Is it cheaper in some senses, more robust? Those types of questions are asked if you look at design and operating principles. So, and the claim in this particular area is that if you do not understand those principles, you are on very thin ice when you go out and design new bugs that, you know, clean up spills or do other things to organisms where you don't really know what the consequences are. So goal 2 is disease simulators, and you all have seen flight simulators, and I'm sure you are glad not to be in that particular machine right now, especially people from London. And but flight simulators have been very, very successful in training pilots and checking out normal situations and checking out situations that hopefully you will never encounter. You know, both the engines fall down, what are you going to do? So the question is, can we come up with a disease simulator? So here you would have a big system with many, many equations and all kinds of things in there. And you can actually personalize these models by taking biomarker measurements of a particular person, you know, blood pressure, gene expression, all these things, put them into the thing, and then try out what different treatments might do. Now you say we are far away from that. Well, actually not. There are companies like Entelos in California that produce these types of models and sell them to Johnson & Johnson. And so, you know, we are getting there. It's not perfect, but it's getting there. So does a disease simulator offer deep insights? Well, you don't get insights into design principles, but you get other types of insights. Well, if I give this treatment, this is going to happen. So if you want to work on a system simulator, then you need lots of data, of course, and you need very detailed computational models. And then you want to account for primary effects and secondary effects, especially, you know, immune responses that come up if you treat cancer, for instance. And there are many challenges, and so we are working toward that, and I would imagine that 20, 30 years from now, if you have some uncharacterized illness, you go to the doctor and things will happen like they happen in car garages. You know, the doctor will hook you up to some computer, and the computer will say, like in the car, well, the sensor is broken, and you didn't even know the sensor existed, let alone what it did. And isn't that true? And so somebody still has to fix the sensor. And so the physicians will not be out of work, but the diagnostic part will be supported by these types of computer programs. Okay, now to my examples. So the first one has to do with reactive oxygen species, or ROS as we say lovingly. And of course they are involved in inflammation and all kinds of diseases. And they're also involved in physiological signaling. So oxidative species usually get a bad rap because you say, oh, they're bad, they're eating up your membranes and all that. Well, they have a physiological function, so you cannot really, eradicate them, or you shouldn't eradicate them. Now, this enzyme NADPH oxidase, or Nox, as we say, is the major contributor to reactive oxygen species. So those are the two things we want to look at, and here's an example, or here's a diagram where you see, the NADPH oxidase is producing these reactive oxygen species, and they go into all kinds of inflammation responses and mitochondrial responses, all that stuff, and there are all kinds of feedbacks, positive, negative, and so on. So our rationale for even getting into that was that we have a good colleague, Han-Jung Zhou at Emory, and he is interested in bone morphogenic protein, or BMP4. And so he was interested in what BMP4 is doing in mechanosensing in arteries. And so what's happening here is you have blood that's flowing through vessels, and what the vessel cells, the endothelial cells, really like is the laminar flow of the stuff. If there's no flow, they're unhappy. If the flow is perturbed, they're unhappy. So they want this, nice, like what's the silent river there or something at Disney World, so where you sort of float along, okay? Because if you have perturbances, for instance, on branches of these arteries, that's where plaques form, and that's where you get into trouble with cholesterol and those things. So he wants to know that, and so we put a system together. Here's our system, and you can see the BMP4 is up there in several places. There's a precursor and there's a mature one, and here's our NADP oxidase. And in the end, the things that are of importance to clinicians are monocyte adhesion and plaque formation and those things. So here we have the OSS is an oscillatory shear stress. So instead of perturbed chaotic stress, what they do is they flush the blood back and forth. And the cells really don't like that. Whereas the ULS is a unidirectional laminar shear. So that's just flowing. That's what cells like. Anyway, so the details are actually not all that important. But this is the diagram that we put together. And he says, can you tell us what BMP4 is doing? So, and then we looked at this NADPH. I come back to that. So then we look at this NADPH oxidase, the Nox, and you look, whoa, so you focus in, you have another mess on your hands. So it's déjà vu all over again. In systems biology, you have all these puzzle pieces that you want to put together, and then you look at one and said, okay, this is interesting for some reason. Let me look at that, and boy, wouldn't you know it, then you have inside this piece, you have that. And you say, how can we even get started? So here's our system, how can we get started? And that is a very interesting question, because nature has not given us instruction manuals. So we have these arrows that show something or the other, some signal or some positive effect, some negative effect. We have dashed arrows, and we really do not know what the functions are that describe these types of processes. So what can we do? So it's a challenge. So it's a challenge that's related to the complexity of biological systems. And, generically, there are large numbers of components. I don't have to tell you about that. Large numbers of processes. That is clear. And then the processes are nonlinear. And because they are nonlinear, you have things where quantitative changes in parameters can cause qualitative changes in responses. So to make it simple, if you imagine you go to the beach and you get some, you're exposed to the sun, you get some suntan. Now you stay a little longer, and the process is exactly the same, but you end up with a sunburn. So you have a slight change in the quantity of sun exposure, and the responses are very, very different. So what's the problem with nonlinearities? Here is the highway net of the United States, and the Department of Transportation is not necessarily known for their PhDs. And if something happens, they will be very happy to give you a detour and say, this is how you get around some blockage in this road. Here's another example from Taiwan. I like that. So there's a hot spring, and everybody tries to get some hot water out of this little hot spring there. So it looks very complicated, but in reality, it's actually pretty simple. So you could go to the house and see which pipe goes where, and then you find what's wrong. So if you have a linear system like this, there may be many components, maybe there are many processes, but it's linear, it's all superposition, you can figure it out. On the other hand, if you look at something like this, so here's a nonlinear system. So let's say these are two genes, X1 and X2, and they affect directly or indirectly their expression. And so the green arrows are positive interaction. The red one is a feedback. It's a negative inhibition. And on the right hand under B, we have a way of writing down equations for that, and now we can play around with it. For instance, if alpha is 0.9 and H is 1, and we kick it a little bit, then X1 goes up, comes back down, X2 reacts, everything is fine. If we change the alpha a little bit, you can see the axis, the y-axis is different, but the response is not all that different. Now, if we set alpha 0.9 and H 0.02, you see different things happening. Now all of a sudden we have a damped oscillation. And this damping, it's over in about an hour in this case. But you say, oh, bad things can happen. In fact, if you go to 1.02, so here's a change of 2%, you can see that the behavior becomes different. So all of a sudden you have what we call a limit cycle. So this oscillation goes on until you kick it back into a different domain. So what this shows is that even very small systems can have surprising repertoires of responses. And that is, for our mind, is very difficult to comprehend. So we need to capture that complexity in some efficient manner. And the question is, how do we do this? So you say, well, why don't you use true functions, or why don't you use linear functions? And because if it's not linear, then you have a lot of possibilities. So why don't we use true functions for these types of systems? Now here's the bread and butter of biochemistry. So you have A plus B, and then goes into P plus Q. So you have glucose and ATP, and out comes glucose 6-phosphate and ADP. Okay. Now that shouldn't be so hard. Now if you ask Mr. Schultz, who wrote a whole book about it, he says, well, it ain't that easy because you have an enzyme involved, and so the enzyme has to hook up to A, and then you have to get the B in there, and then you have to get the A off, and then all that. So if you write it all down, this is what it looks like. Okay? Now, you can say, I do cloud computing, or we have an ATA computer in my institution, so that shouldn't be so hard. Now, that's not the point. The point is that if you want to describe it right, you have to identify all the num and the coef and the constants of this, okay? And that is essentially impossible. And you can imagine if you now have 500 reactions of that sort, the whole thing becomes pretty much infeasible. So that's not a good idea. Why do we not use linear functions? Now, engineering has been extremely successful with linear functions and designing things that behave linearly. And so because of that, we can send a little gadget to Mars that spits out a little rover that sends back pictures. Never tried out before, it works the first time around. It's all based on linear mathematics, much of it at least. So let's say we want to model a heartbeat here, and it's a system of two linear differential equations, a sine function, all right. And you perturb it at that arrow, and you can see the moment you perturb it, the oscillation becomes different. So the amplitude is different. So in other words, if you would run up the stairs, your heartbeat goes up, it never comes back down until you kick it back in some way. That's not good. On the other hand, if you look at this very, very old model from the 1926, I think, from Vanderpoel, it's a nonlinear oscillator. You kick it, it goes up, responds, adapts, comes back. So we need nonlinear for these types of behaviors. So the way we do it is to use biochemical system theory, and I'm not going to say much about it. I have a couple slides here. So we look at boxes, and inside are X's, and the X's can be metabolites, or they can be gene expression levels, or they can be something that has nothing to do with biochemistry. And then we have fluxes going in and fluxes going out. And we can immediately write down equations, like, here's our input, there's our output. And what do we know about these functions? Well, these functions are probably functions of something that's inside and something that's outside the model. What exactly it is, it's a different question. The only thing that we really, really know is that these functions are complicated. So what can we do about it? Well, we use an approximation with a power function, something that only a mother can love, but it's actually very useful. So once you get used to this, it's a useful presentation of these types of unknown functions. It's an approximation that is nonlinear, and it has certain advantages. One significant advantage is that we can map the structure of the system onto the parameters back and forth in both directions. What does that mean? Well, here's a linear pathway. It could be a biochemical pathway. It could be a food chain. It could be a car production process. It doesn't really matter. We know immediately how to write down equations for that. And we want to compare it with a system that's the same, except it has a feedback. Okay. So we know that from the structure of these biochemical system theory equations that we have x1 in there, because x1 is a substrate for this process. We have a rate constant alpha 2, which describes the turnover of that process. And we have what we call a kinetic order, this G21, that describes how strong the effect of x1 is on this process. Okay. In the bottom, we can do the same thing. Now we have two variables. We have the x1 again, and we also have the x4, which is an inhibitor. The theory tells us immediately that the kinetic order of this inhibitor has to be negative because it's an inhibitor. Furthermore, the more this parameter is away from zero, the stronger the effect is. The closer it is to zero, the less substantial the effect is. So we know that this parameter is less or equal to zero. If it becomes zero, actually, then you see that the formulation becomes the formulation on top, the pathway becomes the pathway on top. What does that mean? If we can estimate parameters, and if we estimate that the G41 is something like minus 0.1, 0.5, we know that there's some inhibition signal. Whereas if the parameter is minus 0.02, we can say the inhibition, maybe it's there, but if so, it's not very substantial. So it's a very significant advantage because we can write down equations immediately without knowing much more about the system. The hard part is to find the parameter values, and we have heard that many times here, and I'll come back to that with the examples. So there are all kinds of applications if you're interested in that, metabolic pathway, signaling things. We've been working on neurodegenerative diseases lately, in Parkinson's, schizophrenia, gene circuitry, other things. So back to our BMP4 system. So I asked the question, how can we even get started? And the answer I want to give is, well, we can use BST as an initial default. Now, there are many people in the world who say, well, your functions are a problem because they don't do this or they do that. To which I say, if you know better, do something else. But if you don't, this is a good way to get started. Okay? So for instance, if you imagine you have a receptor and ligand is binding to it, the receptor kicks on a cascade, the cascade goes down and turns on some gene. How in the world are you going to describe it mechanistically? It's just, it's not feasible. And so to get started, these power law equations are actually quite nice. So we can take something like this and write it down immediately. A computer can do it. So here it is. Now, if you have keen eyes and you're still awake, then you will see that there are some t minus taus. There they are. And you can actually approximate these types of delays. So these are delayed reactions. You can actually approximate them very, very nicely with equations that look like BST equations and that do not require any additional parameters except for this tau. Okay, so you can do that, and you have auxiliary equations that run in the background and that allow you to model these types of delays. Now here's a parameter estimation. That's always difficult, and we have heard it. And here in this particular case, we looked at steady state solutions, and we have three different situations. We have something normal, we have something with a laminar stress, and we have something with an oscillatory stress. And if the system equilibrates, comes to a steady state, then that allows us to at least constrain some of the parameters. For instance, we look at this equation x4 dot. In steady state, the x4 dot has to be equal to 0, so the two gamma terms have to be the same. And now if we have three scenarios, you can see you get three linear equations as soon as you take logarithms of these two gamma x raised to g terms. Everything becomes linear. So now we have three equations. We have, if you count the gamma 7 over gamma 5 as one parameter, then we have three parameters which we can sort of estimate from that. Now this is a lucky guess, not a lucky guess, it's a lucky situation because we can do it. In many other cases, it's not so easy. Here are some results, how the BMP4 precursors and mature and so on react to different stresses, and the details are not important, but you can fill in the blanks, and you can figure out what parts in between the system are doing, and you can test different scenarios. Here, we don't need that. So, we have successfully converted this biological diagram plus heterogeneous information. So if you have information that says, this thing is actually affecting that process, you know where to put it into these equations. The pieces of the puzzle fit together in this case. The model fills in gaps, so we can reproduce time courses of things that were not measured. And it demonstrates, that laminar stress is protective and that oscillatory stress is bad, is pro-inflammatory. It also shows that these time delays are actually crucial. If you take them out, then things fall apart, which is interesting detail. So now we want to zoom in on Nox1. It's right there in the middle, and see what that looks like. So if you look at active Nox1, and we're using now vascular smooth muscle cells because there's more information available. So the active Nox1 is in that ellipse, that orange ellipse. There's a lot known about the assembly of Nox1. So to make Nox1 active, you have to have four components. You have to have this P47 FOX, and it has to be phosphorylated. You need this RAC1 GTP, that is phosphorylated. This is the GTP, this is the GDP. And we need this Nox1 activator, which is not phosphorylated. So this form is phosphorylated. And then we need this thing. So there are these four components. They have to come together, and that activates Nox1. And then Nox1 does what it does to oxidative species. The question we asked was, is it important how the thing is disassembled? We know how it's assembled. Is it important how it's disassembled? And why is that an interesting question? Well, if you imagine that you take, first of all, you could just do proteolysis, so destroy that thing and build it new. And we'll look at that. The other options are F2, F3, F4. Now let's look at F4. In F4, you recoup this part here. You recoup the GDP form of RAC, and you have the phosphorylated P47, and you have the Nox1. So you get this part, that part, and this one. Whereas if you disassemble it this way with F2, you get the phosphorylated form here, you get the phosphorylated form there, this one, and you get the this form here. And the third one is similarly. So, depending on how you take this thing apart, where you take the phosphate off, you go back to slightly different states. Okay? And the question is that important? So, there are some reversible reactions. Here's a reversible reaction, here's one, here's one. There are regulatory signals, and that we count S1, call S1, S2, S3. For instance, there's the hormone angiotensin that is working on S1 and probably S2. There's protein kinase A, and then there's this PI3K, which works over here in the signal. And then we have 3 recycling pathways that I showed, 2, 3, 4. And we have one pathway that just destroys that thing. It's gone. So I think I said all that. So here's the proteolysis, here's recycling, but the recycling pathways lead to different states. And the question is, how can you set up the system such that it's cocked for the next event, for the next signal? How can we get it ready in a short period of time? So the question that we want to ask is, which mechanism of Nox1 disassembly are important? We know what the assembly looks like. Is recycling more important or less important, is equal to proteolysis? How do these disassembly processes affect the responsiveness of the system? And is there one prominent recycling flux and the others are negative? And does it even matter? For instance, does it matter whether the FOX and RAC and so on in their active state or in their inactive state to be ready? Does it matter? So we set up a model. It's a mass action model in this case, which happens to be a special case of general mass action system in BSD. Here we did mass action because everything we want to do is sort of relative and your order of magnitude. We have 8 dependent variables, 3 input signals, 16 unknown fluxes rate constants. We have 8 linearly independent parameters, so you can, if you look at the steady state operation, you can throw out some of the parameters. But there's little really crisp information on the rates and on other parameters, which is the typical situation and not the exception. So what we really want to do is create a model that only minimally depends on specific parameter values. So it's not the goal to find a solution that says alpha has to be 1.221. That's not the point. We want to know order of magnitude. Is this better than that? Is it much better than that? That type of question. So what we did was we used the Monte Carlo simulation with filtering against appropriate criteria of effectiveness. So what does that mean? So we use a parameter space for these variables. And in the end, if we started out by saying, is the proteolytic flux, is it as important as the recycling fluxes altogether? Let's put them all together and let's see whether those two are equally important or not. So if you like, we have flat priors in logs, so we look at the flux rates, and in biochemistry everything is logs, so we take logs. We start with 20,000 in samples, and then we did some resampling and so on. And then we looked at system instantiations, so parameter sets, value sets, that satisfy 2 criteria, 2 sets of criteria. The first type of criteria has to do with experimental observations. For instance, there are data on the expression of Nox1 protein in response to angiotensin, this hormone that I mentioned. There is something about the regulation of Nox1, and there's something about the regulation by PMA, which is an activator of PKC, protein kinase C. The second set of criteria is different in a way. So we talk about a ready-to-respond state. So we say we want to return the system to a state where it can respond as effectively as possible. And what does that state look like? So this state should give you a maximal responsiveness to signals, and it gives you a minimal response time, at least on a coarse level. So if we look at checking against the experimental criteria, then on the top left, we have screening just against criterion 1. Here we have criterion 2, 3, and 3, and here is all the criteria together. And you can see that in this particular case, at least, this strategy worked really, really well. Because what it tells us is not recycling flux has to be 1.25 nanomolar per minute. But what it says is the recycling fluxes, all combined, all three combined, must at least be 2 to 3 orders of magnitude larger than this recycling flux. Okay, so that's what this says, because here the proteolytic flux is something like minus 1.5 in log 10. Here we have something like 1 to 2 in log 10, so you have your 2 to 3 orders of magnitude. That's a very strong result, because we don't know much about these parameters at all. And it says, you can only satisfy this experimental criteria if you do this. In fact, we looked at the other criteria too, and we get sort of similar results, and I'm not going to go into that. So here it is again in a different representation. So here the proteolytic fluxes, here the recycling fluxes, they are clearly separated here. That's luck. I mean, you cannot expect this. We were lucky, okay? Okay. Luck favors the prepared, I guess. That's what they say. Okay, so here are the 3 recycling fluxes, and you say, well, there we cannot really say much about it. Maybe this should be a little higher, and this may be a little lower, but it's hard to say. So we cannot say that. So here's the question that I asked earlier. Should the subunits of this Nox1 normally reside in an active or in the inactive forms? Okay. And I challenge you to write down yes or no, or inactive, active, and then you can check against what I'm going to tell you. So we're going to start with a little thought experiment. And instead of having 4 subunits, we only have 2 subunits. And to make it easy, they can be either on or off and nothing in between. And then we do it right, just to see whether this even makes sense. So let's say we have this one, the P47. It can either be active or it can be inactive. And we have the rack, and the rack can be active or inactive. These 2 other things we forget about them. Now you can see if both are over here, or let's start here. If both are in the active form, these are the active forms. They're both in the active form. What happens? Well, you constantly produce Nox1. Okay, so the system is not responsive to signals because it's doing its little thing no matter what. You can't stop it. So that's not a good solution. Let's say they are both outside. Now you only get signal 1. What's happening? Well, signal 1 activates this guy here, but because you cannot go over here, it's always in this form on the right, you're not responsive to S1 alone. You're only responsive to S1 and S2 if they happen to come both. Okay? So you can see from this that it really does matter whether you are in the active form or the inactive form. Now, in nature, nothing is black or white, so everything is sort of, So we need to figure that out. So I said already what's in this table, and so if, in these extreme cases, you really have problems. Now we want to do it, so it's important, and how do we address it? So we looked at the possible modules of deactivation, disassembly, leaving out this proteolytic flux, which is very small. So we can either have just F2 or just F3 or just F4, which is 2, 3, 4, or we can have combinations of 2, or we can have all 3. And what do we do with it? Well, we do another Monte Carlo simulation, and we check against the responsiveness to angiotensin, which is represented by a combined signal to S1 and S2. Now, I'm not going to go through the results in detail, but here's sort of a summary of the results. The dominance of these particular flux patterns depends on the rates of these reversible reactions. In particular, Now this will probably surprise you. You need slow reversible reactions. Reversible reactions are this, this, and this pair. Okay? Why do you need slow reactions? Well, if you have very fast reactions in comparison to other things, everything always equilibrates. Okay, you have no controllability of that thing. No matter what you do, you hit it with a signal. It says, "I don't care. I go back, and I'm back to my equilibrated state." Furthermore, this relationship between F9 and F10 at steady state, that's the relationship between this flux and that flux, is important. Otherwise, you cannot control that thing. So there they are. Okay. And then we looked at response times. So how long does each of these modules need before you're ready for the next signal? And you see there are tremendous differences. So module 1 is very slow. 2, 3, 6 are very fast. Here's the on response, here's the off response. What does that mean? Your system sitting in its resting state, you hit it with a signal, how fast does it respond? And now the off response, the signal is gone, how long does it take before it goes back? So you can see this analysis shows you that module 2, 3, and 6, that is 3, 4, and 3 and 4, but not 2. So 2 we don't want, 3 and 4, or both of them, that's what we want. So those are dominant fluxes that re-establish this ready-to-respond state in a short period of time. So here they are. So the system has to be in its inactive states, predominantly, not totally, but predominantly. You want this going in this direction. And then if you have signals, and then you're responsive to these things. You can now take the system and study repeated signals and signal trains, long signals, short signals, all those things. So, I'm coming to the end. Here's summary part 1. If we look at this Nox1 disassembly, and we have done some dynamic analysis, but I didn't have time to show that. So the pathways of disassembly are not equally important. Now, after what I told you, you're not surprised. But when we talked with our experimental collaborators, they had this blank stare. I said, why? Why do you want to disassemble this? We want to assemble. They said, yeah, you cannot only assemble, you need to go back to be reactive. So even the question to people who are not used to these types of abstract thoughts, the question even is complicated sometimes. So be forewarned if you talk with people like that. Proteolysis is inefficient, which shouldn't surprise us because you take this protein, you destroy it, and then you have to build it up again. So that's not, so reuse, recycle, reduce, reuse, recycle. The thing is, they do that too. Now in this case, yeah, you should have some proteolysis probably, but it should be at most 1% of the recycling fluxes, more like 0.01%. The optimal recycling depends on relatively slow reversible reactions. If everything is very fast, you cannot control it. It's doing what it wants, and it's out of control. And there are certain numerical relationships, and the numerical relationships is, again, it's not 3.11. It's this has to be much bigger than that, this has to be much smaller than that, those types of relationships. And they are sufficient to characterize this. In a way, that's a much stronger statement than if I say, here is the set of parameter values that fit the thing. The subunits P47 FOX and RAC1 should reside in their inactive dephosphorylated forms. And in fact, there are some papers, once you find something out, then you go to the literature and somebody says, yeah, these are in inactive form. Why is that? They should be ready to go. No, they shouldn't. They should be in inactive forms for a reason that I showed you. But there is some experimental evidence that that is true. The optimal ready-to-respond state actually turns out to be very noise-resistant because you're in these inactive states. So if you have a little bit of signal, it doesn't do much. You go back to your inactive state, and only if you really get big signals, then it happens. And the analysis of repeated signals actually suggests that there is regulation in there. So if you always get signal S1, the system gets used to it. It says, aha, I'm ready for S1. And it seems, and we don't know what it seems that that is somehow regulated so that you go into some recycling pathways rather than others. But nobody knows experimentally, and we haven't figured it out. More generally, if you want to do pathway modeling or any sort of modeling, it's often very beneficial to have default models at least to get started. Now, many people use mass action systems. You do mass action, and that's your default, fine. We do these power law systems because they are a little bit more general. They are excellent tools to get started. They are not the solution to all world problems. Otherwise, we would have gotten MacArthur Awards or something. But they are not. But they are good ways to get started. Parameter estimation is always an issue, and we are getting more and more away from the point estimates and look at these, scan the parameter spaces and look for good areas and bad areas and try to figure out what that means. The models lead to computational structure that fill in the blanks. So we can make statements about things that are not measurable or that have not been measured. Because of that, we can suggest testable hypotheses. We say, if you do this and that, that should happen. Try it out. The model can reveal relatively complicated insights into these systems, for instance, with the inactive states, active states, those type of things. The mid-sized models, I said in the beginning, if you want to prove stuff, you need very small models. If you want to do disease simulation, you need these huge models. And we are all dealing with mid-sized models. That's the way to go, at least to get started. And then you can abstract them and simplify them and look for design principles, or you can expand them and make them into disease modulators, simulators. This is Wei Wei Yin, and if things go her way, on Monday she will be Dr. Wei Wei Yin. So, wish her luck. And those two are responsible for whether she will succeed or not. We have been blessed with support from various agencies, so that was good. There's my website, and I want to finish with a little infomercial. Here it is. This is the new baby. Okay, came out a couple of months ago. It's a real introduction for undergraduates who had some biology, a little bit of computing, or early graduate students. Here's something at 360 exercises, and we had to solve them all, and don't tell your students, but the whole thing is about this big if you do all the solutions. But as instructor, you get the solutions. You can actually rent the thing for 6 months or 12 months. So if you want to look at it, I'll be happy to show it to you. Thank you.
Thank you. So that was a very nice summary and gave us a lot of good examples of the effects of nonlinearity. Are there any questions?
Yeah.
So one question, like what you at the beginning you said, sometimes when the parameter changes by 20%, the quality is changed. And how to deal with this parameter identification? Because the measurement can easily change by 20%.
Well, my students always make fun of me because I answer questions with "that depends." So let me do that. Let me say that depends. It depends. It really depends on the system, on the size of the system, the structure. There are almost always parameters that are very insensitive. You can change it by 200% and not much changes. And then there are parameters and you change it by a few percent and it makes a big difference. So what you want to do is identify the latter or distinguish, classify them into very sensitive and not, because the insensitive parameters you don't really have to measure very precisely. It doesn't matter whether it's 1.2 or 1.5 or 2. But the parameters that have a high sensitivity, those you have to look into and see that you do the best you can identifying them. But if you do, what I showed here with these, bigger spaces, if you can do it, if you can afford to do it, then you are probably better off.
Is the model specification is too sensitive?
It can be that the model is very sensitive. So you are close to a bifurcation point and you change this parameter and something goes from a stable point to an unstable point surrounded by the limit cycle. And we all know that. We can construct examples where you change it by an epsilon and things fall apart. It can be that you have the wrong model structure. We had a model that was very sensitive, this purine model for that matter, because we intentionally ignored a very secondary pathway. Well, under certain conditions, you didn't have this pathway and everything accumulated and the whole thing would blow up. So it's really hard to say in general.
Okay, well thank you.
Thank you.
Didn't fall behind.